Good morning. Today I'd like to talk about sperm DNA fragmentation and its relationship to IVF. Is it important? The video was prompted by a recent publication in July 2025 from some Chinese authors suggesting that elevated sperm DNA did not significantly impact the IVF outcomes, suggesting limited utility as a routine pre-treatment in ART. This paper suggests that the sperm DNA fragmentation testing adds little to IVF management and does not need testing. However, I think there's more to this story, as I'd like to demonstrate in this video. But by way of welcome and disclaimer, my name is James Danger. I have a PhD in embryology, and I've been working in IVF for more than 50 years, before Louise Brown. These videos from my Fertile YouTube channel are my attempt to bridge the gap between information that you may need for your fertility treatment and the most recent publications available to your clinic staff. Now, I'm not a medical person, but this information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor and maybe to help in understanding the advice that you receive. I hope it helps. Okay, before I go much further, I always allocate this page to potential supporters of my IVF daily video channel. I'm just starting out, but if you want to promote your company or clinic or product, this just may be the place for you to consider. It's worthwhile noting that in supporting my IVF daily channel, your support may mean an IVF client becoming a better informed IVF client. You can click subscribe or become a sponsor, or just make a donation to help others in their fertility journey. Okay. So why discuss sperm DNA fragmentation? Should we test it or just forget about it? Well, in most of IVF's history, all swimming sperm were just seen as being the same. Embryo development was always considered to be part of a function of egg health, and the sperm really, as long as it was swimming, it was okay. But we always knew right at the very beginning, back when I got involved in IVF around about 1982, that about 10% of cases with poor sperm quality fail to fertilize after IVF. The microinjection of sperm, called ICSI, when that was developed to overcome this problem, sperm became very much commoditized, where they just seem to be a cell that you select, inject, and forget. There's no QC and there's no rules about how you do this. However, sperm DNA fragmentation had been tested for some time, but there was still mixed thinking about whether it was a worthwhile diagnostic technique and whether it was a marker in the preliminary management of IVF treatment. But you fast forward to 2025, and now the quality and the selection of sperm are increasingly seen to be important, and embryo development is now seen as a product of the quality of both the sperm and the egg. So what's changed? I'd like to quote from Dr. John Aiken's paper in Andrology in 2023, where he says, in considering the clinical impact of sperm DNA damage, the male gamete cannot be considered in isolation, because of the clinical consequences of such damage may also depend upon the ability of the oocyte to detect and repair genetic or epigenetic defects in the fertilizing spermatozoon. The involvement of both male and female gametes in determining the consequences of sperm DNA damage for the embryo has been captured by what's called post-meiotic oocyte collusion hypothesis, which sets out to explain how DNA damage in the sperm may ultimately translate into an increased mutational load in the progeny. So this is all going on in a slightly different direction, but he's actually raising the really important issue, which I'll get all the way through this video, that the role of sperm DNA competency is a critical thing in embryo development and your success in IVF treatment. So when you or your partner's asked to have a sperm test, basically you produce a sample into a jar, and the scientists will look to see how many sperm there are, the concentration, how well they're swimming, its motility, and what they look like. You have a nice normal round head and a long normal tail, morphology. But when you do look at DNA damage, you're also looking at something healthy, something about the health of the sperm. And there's a lot of divided opinion on the value of this. I may also point out that there are no quality assurance control programs for demonstrating competency, and there are a lot of different techniques for looking at sperm DNA, which I'll allude to. And this is half of the problem, that the issue of assessment is very diverse. So what is sperm DNA fragmentation? Well, as we know, this is what a sperm looks like, a little head and a tail, and all the chromosomes in the sperm head, which normally look like this, however, have been modified for the sperm to protect it. These sperm samples are highly compressed and wrapped up a bit like a golf ball, where the chromosomes are squeezed very, very tight and protected with a layer of protamines to allow the sperm not to be damaged in its long journey from the testis to the site of fertilization. But a lot of the damage to sperm DNA occurs before the spermatozoon takes on this shape, and therefore you can have sperm which has some damaged DNA, which has then been wrapped up, and it's not unprocessed until after fertilization. Therefore, in a sample, you may have some sperm with DNA damage, while others are quite healthy, and therefore you have various amounts of DNA damage in the sperm. Rarely do you have a sample where all the sperm are healthy, and almost never all the sperm are damaged. There's lots of different tests for doing this, but in reality, most healthy samples have less than 5% damaged sperms, usually only 1 or 2 or 3%. But in really stressed males, you have rates of 30, 40, 50%, and they're often related to other sperm parameters like morphology and motility. And we're talking about damage. You can see here the damage. You can have little nicks here and there. You can have a single strand break, which still has one normal strand of DNA and a broken one. And you can also have other more critical ones where the damage has been on both sides of the DNA, which are a lot harder to repair. So when you're testing for it, there's lots of different tests. But in this diffusion test, the sperm head swells up and you stand the chromosomes. They form those nice little blobs. But when it's unhealthy, they fail to unwrap correctly, and you get these little, very condensed heads. Maybe worthwhile pointing out that in natural conception and with many aspects of IVF and ICSI, many of the sperm with damaged DNA are filtered out because of the other factors such as motility and morphology exclude them from getting to the site of fertilization or achieving fertilization. Regardless, though, if you have a sample with high levels of sperm DNA damage, that's a marker of men's health, and male reproductive health is related to male health overall, so high levels of coronary disease and diabetes are linked to male reproductive health, and measuring the DNA damage is a tool for that. Most of the damage is oxidative damage in the testis, and there may be some lifestyle issues that people can look at and discuss it with your doctor. But the real problem is there are lots of different tests for different sorts of damage, and the whole area is somewhat confusing. So to summarize the clinical status current status in clinical management, most of the industry thinks it's not important. Very complex studies cannot confirm an elevated sperm DNA fragmentation is linked to pregnancy. Therefore, there's no need to do the test. And anyway, sperm samples are highly processed, and a single sperm taken for rejection almost makes the testing redundant. And anyway, transferring the best embryo, which is nearly always reflecting an embryo created with good quality sperm, is the reality of IVF and may mark the importance of subgroups. But there are other people in the industry who consider that sperm DNA fragmentation is important. It's related to other aspects like motility, morphology, and concentration. It may be related to male health, but also related to age. And I'll show you, the older the guy is, the more DNA damage there is. But there is quite a lot of data which I'll present suggesting it's linked to poor IVF outcomes. It may be linked to miscarriages. And here's the critical thing is, I think it only becomes important as female age increases. Okay, so let's look at some of the publications where they consider that sperm DNA fragmentation is of a little value. Here's one published in 2018 from Taiwanese authors. And they showed that they looked at the fertilization rate, the number of good embryos and pregnancy in samples. There's a neat sample where there's lots of damage or low damage, and they found there was no difference. They did note that decreasing motility was associated with high fragmentation values, suggesting that the DNA damage is impacting on the motility. And when they tried to do a receiver operating characteristic analysis, they identified a cutoff of 20% as being the one where any effect may have been important, but even at that, they could not find it was related to IVF outcomes. So they concluded that the fragmentation score did not provide independent information regarding fertilisation, embryo quality or pregnancy for infertile couples who receive IVF and ICSI, but were consistent with some semen analysis qualities regardless of IVF outcome. So you measure it, but it's only reproducing information received elsewhere. Here's another one by a similar group, but updated to 2025, where they also reported no difference between above and below 30% DNA fragmentation. They also looked in this paper looking at the babies born and found that there was no difference in the health and congenital malformation of babies born. And they did show that male age was related to sperm DNA fragmentation. So they concluded that elevated sperm DNA fragmentation did not significantly impact IVF outcomes, suggesting limited utility as a routine pre-treatment assessment in IVF. But if we're talking about eggs, how important is this? This is an article published in 2019 looking at egg quality, where they used donated eggs from young women, and they found that the fragmentation did not significantly impact the outcome of IVF cycles using eggs that had been donated from young women. Even in cases of advanced paternal age that had high levels of damage, still resulted in normal outcomes. And they concluded that the fragmentation was not significantly correlated with pregnancy and oocyte donation cycles, therefore why bother testing it? However, giving a whiff of some impact though, they did make the observation that they had more good quality blastocysts when the sperm DNA fragmentation was low. In other words, low DNA fragmentation produces more good quality embryos, and more good quality embryos are related to your chance of pregnancy. So here's now a couple of articles looking at meta-analysis where instead of looking at a single paper, these authors have looked at lots of papers and tried to make some overall assessment. Here's one that was done in 2023 by some Japanese authors, where they looked at lots of reviews and they did a meta-analysis looking for an association between individuals with high sperm DNA fragmentation. I like this MAMAB, which is individual assigned male at birth, and adverse outcomes. And they could show that even though there did appear to be some association with live birth rates and pregnancies following ICSI, the results are inconclusive. And they make a point. There are a lot of disparities between assays, inconsistent reference ranges, etc., making this whole area difficult to do. So the point you want to get from this is that it's very hard to do a meta-analysis if the base standards of identification are all over the place. Here's another one published March last year, an umbrella review. I think these were Egyptian authors. They looked at 50,000 cases, 22 reviews, of which 15 were meta-analysis, and they concluded that overall there was the association between sperm DNA fragmentation and live birth was weak at best, and that related to pregnancy, miscarriage, implantation, and fertilization was also weak. And it was also very weak or non-significant for doing the type of investigation, either IUI, IVF, or ICSI. And they concluded this umbrella review did not find any convincing or suggestive evidence linking sperm DNA fragmentation to IVF outcomes. And they made the point that any clinics making claims that it's of value should be treated with caution. So, if you, on the hand, take the other view that sperm DNA fragmentation may be important, you could say we know it's related to morphology and motility. We know it's related to male health and age, but they're still unsure about whether it's linked to poor IVF outcomes, whether it's linked to miscarriages, and how important is female age. Okay, let's look at a couple of bits of data. Here's the first one, was published in February this year, looking at the rate at which embryos divide. This is the embryo morphokinetic data, and they concluded that sperm DNA fragmentation could adversely affect the rate of usable blastocysts on day 5 and delay the formation of blastocysts, that is, embryos are dividing slower and fewer will get to endpoint, but only when the oocytes are derived from women of advanced paternal age. then their results suggest that oocytes derived from younger females have a greater capacity to repair sperm DNA fragmentation than from women of advanced maternal age. Here's another one looking at sperm selection. This is a bit busy, I apologise. This was a paper published in Fertility and Sterility in May this year, looking at the impact of how you prepare the sperm using microfluidic sperm selectors. Instead of doing sperm washing and letting sperm swim up, you use a device which separates out highly active motile sperm. This one's a Zymot, but there are lots of other ones, probably better ones, I don't know. But they identified that the fragmentation was a lot lower when you use these devices than the swim up, than analysing the oocytes. Using these two different sorts of preparation, they showed a significantly higher rate of blastocysts per egg injected. This was not observed with donated eggs because basically all the young eggs are healthy. So embryos from swim-up group, this is this group here, showed a significant delay in the time of the pronuclear appearance, that is how long it takes for the sperm to be processed, and the formation of the morula. And so this study provided valuable information on the use of selection devices for a non-invasive sperm selection. When these type of devices were used, the authors observed a reduction in DNA fragmentation in the sample and an enhancement in the number of usable embryos available to the clients. These findings could be compatible with a reduced capacity to repair sperm damage due to poor oocyte quality caused by advanced age. Okay, I'd like to look at a couple of papers looking at cumulative pregnancy rates. If we're talking about the number of embryos available for transfer, the more embryos you have available, the more embryos can be either transferred or frozen, put back as FET. And if you look at the cumulative pregnancy rate, that's when all the embryos have been brought back, how many people are still being pregnant. So that's a chance of pregnancy per pickup. These people, these authors in China, I believe, in ICSI cases, the cumulative live birth rate decreased significantly as the sperm DNA fragmentation increased. And these effects were stronger with the increase in female age. They found this a similar pattern for sperm DNA fragmentation and female age was related to for IVF, but the correlation was not significant. So they concluded the sperm DNA fragmentation had a significant effect on the cumulative probability of achieving a pregnancy using ICSI. And it could well be that in this case, the using IVF, and only the sperm which have good quality are able to fertilise in IVF. And there's a bit of confusion about the difference between IVF and ICSI, I have to say. Here's another one looking at unexplained infertility. And here in the fresh embryo transfer, there was a significant difference in the implantation rates, clinical pregnancy rates, and live birth between between samples with low or high levels of sperm DNA fragmentation. However, in the frozen transfers, there was no difference. I think that basically at this point in time, the FETs per FET, the embryos are all basically healthy. Using multivariate logistic regression, sperm DNA fragmentation was an accurate predictor of live birth rate when adjusting for all other compounding factors. And they concluded that high sperm DNA fragmentation was associated with a lower cumulative pregnancy rate and a higher miscarriage rate in ICSI for cycles of couples with unexplained infertility. So we need to talk a little bit about egg numbers and egg quality. So here was a paper published in 2023 looking at egg quality, not so much egg age, but egg quality, and they showed that the lowest fertilization and high quality embryo rates were observed when high sperm DNA fragmentation was associated with the presence of dark cytoplasm vacuoles, etc. So when you had poor quality eggs and high sperm DNA fragmentation, in those cases, it was because the egg was actually of poor quality. The lowest implantation pregnancy rates were observed when a high sperm DNA fragmentation was associated with the presence of abnormal egg cytoplasm, vacuoles, defective perivillin space, and fragmented polar body. The effect of sperm DNA fragmentation on miscarriage rates was also significantly influenced by the presence of oocyte, cytoplasmic granules. They concluded a high sperm DNA fragmentation index increases the likelihood of miscarriage in ICSI cycles, and that effect may be potentially magnified by the presence of oocyte abnormalities. So, once again, looking at oocyte quality and looking at the rate at which embryos divide. In this group, they looked at, in a younger group, there were no differences between high and low sperm fragmentation in the rate of usable blastocysts formed on day 5. However, in the older group, the rate of usable blastocyst formation on day 5 was significantly lower in the high fragmentation group. And they concluded the sperm DNA fragmentation could adversely affect the rate of usable blastocysts on day 5 and delay the formation of usable blastocysts, but only when oocytes were derived from women of advanced age. Here's another article looking at the impact of maternal age on repairing capacity of oocytes on paternal DNA damage, published in July this year. They found there was no significant differences in younger women and patients between 37 and 40. However, when the maternal age was greater than 40, significantly lower embryo and blastocyst quality, blastocyst development, pregnancy implantation rates were observed. Their findings suggest that older oocytes may have a reduced ability to repair sperm DNA damage, as demonstrated by poorer IVF outcomes in couples with a higher sperm fragmentation index and maternal age. And they suggested that novel sperm selection techniques may well be, they listed a few here, seen to isolate less damaged sperm, thus having the potential to decrease the aforementioned adverse effects. So here we're getting to a point here where even though you have high levels of DNA fragmentation and older quality eggs, you can still have better impacts by being how you prepare the sperm for IVF. I've included this one. This was quite a difficult paper to read, but basically they showed that high levels of DNA fragmentation have an impact on ICSI but not IVF. And I think that possibly could be because the oocyte itself is doing some selection of sperm. But the reason I included this paper in the talk was that when all other parameters have been looked at, and you then biopsy the embryo and look for the number of euploid embryos, that's along with the correct number of chromosomes, that was independent. So the moral of the story is that you may have fewer blastocysts, but if you have a blastocyst from a fertilization of an older lady with high DNA fragmented sperm partner, and they get to blastocysts, those blastocysts appear to have the right number of chromosomes. And I'd like to get into a couple of papers looking at pregnancy loss, but once again, I'm not a clinician, and I'm only recording the observations of the authors. Here's one that we're looking at pregnancy loss in terms of sperm DNA fragmentation. And the authors noted that couples that failed to achieve a pregnancy had a high DFI compared to the rest of the cohort. So they concluded at referral, sperm DNA fragmentation, morphology, and concentration cannot be used to identify early repeat pregnancy, recurrent pregnancy loss, couples at risk of another pregnancy loss. Increased baseline DFI was associated with the difficulty of achieving another pregnancy, and improving sperm morphology was associated with increased odds of a live birth. I think you really need to read that paper to make more sense of it. But basically, they're just saying that early pregnancy, recurrent pregnancy loss is associated with sperm DNA fragmentation. Here's another one, published in January 23. They concluded that men within couples affected by recurrent pregnancy loss or infertility have a significantly higher rate of sperm DNA fragmentation compared to couples. However, after adjusting for a lot of covariates, sperm DNA fragmentation was only associated with recurrent pregnancy loss. Now, here's another one published in January 25. They looked at I think these were from Sweden, this group. They found that in ART conceived children, a high level of DNA fragmentation above 20% is associated with increased odds of preterm birth, and a fragmentation level of greater than 10% is associated with an increased odds of preeclampsia and IVF. And these authors made the claim that this finding is of clinical importance, and they do acknowledge the benefits of routinely testing for DFI is still under debate. But their observations suggest that to reduce the risk of preeclampsia, it may be preferable to perform ICSI instead of IVF in the cases where the fragmentation is between 10 and 20%. A DFS-based risk assessment and screening of high-risk groups can be clinically implemented for an individualised risk assessment to reduce the risk of preeclampsia. So once again, this is, I'm not a clinician, and you need to read this paper to understand exactly what the authors are saying. And the last one I'd like to bring to your attention is a publication in August this year looking at pregnancy loss and double-stranded DNA. And basically they're saying if you use a different assay, the COMET assay, and you look specifically for double-stranded DNA, that is where both of the strands have been broken, they concluded that this study of male factor fragmentation in a large cohort of men attending a recurrent miscarriage clinic where they are already focused on clinical investigation shows a strong association with double-stranded sperm DNA fragmentation and male factor-driven miscarriage contribution, highlighting the importance of male investigation in couples experiencing recurrent pregnancy loss. Okay, so what have we learned about sperm DNA fragmentation and IVF? And once again, this is not medical advice. I'm very clear on this. But we can say that there are reports that this may be influenced by maternal age. Eggs from older women may be less able to repair the damaged sperm in time before it needs to start dividing. Thus, sperm DNA fragmentation may impact IVF, but the fertilizing sperm has elevated fragmentation and the oocytes from an older client. So maybe testing may be of value when the age of the client starts, what we say, start to become mature. You may like to use improved sperm selection tools to improve the sperm selection process, and you may like to discuss with your doctor male lifestyle changes to reduce the oxidative state. But I would just like to make one or two comments. So why is this important? Well, we know that genes in the male prenucleus contribute to placental development. Work done in 1964 by Serrani in Cambridge showed that if you took two male pronuclei in a mouse and put it into a mouse egg, you got a placenta, but no embryo development. So this is what's called a hydatidiform mole, and so we know that abnormal sperm DNA may contribute to an abnormal placenta. We know of half the miscarriages are due to chromosomal imbalance, and you can test that using PGTA, but the other half may be genetic or other. Ensuring that the embryo has the healthiest DNA surely should be our goal. And the only thing we can really do at this stage is to use sperm selection devices to minimize the impact. So your clinic may or may not routinely test for sperm DNA fragmentation when they request a sperm sample before treatment. We know that a high sperm DNA fragmentation is a marker of some form of sperm stress, and high values are often associated with poor sperm quality, such as motility or morphology. A high sperm DNA fragmentation index however, are not a good diagnostic predictor of a chance of following pregnancy, mainly because the best embryo is selected for transfer, and this impacts on your pregnancy rate. But there is some data to suggest it may be linked to miscarriage and may impact on adverse pregnancy outcomes. It is linked to poor fertilization rates and delayed embryo development. This may be due to delays in DNA repair in the newly fertilized embryo in older eggs. But by picking the best embryo for transfer, it does not appear to alter your chance of pregnancy. And about this, you need to discuss it with your doctor. So, once again, what can you do? This is not medical advice. But it seems to me that if you are over 35 years of age and your partner's sperm sample is less than perfect, then you may request your clinic for a sperm DNA fragmentation test or inquire if your sperm preparation for IVF ICSI uses a sperm selection device to improve the number of healthy or low fragmented sperm in the sample used for insemination. Actually, the whole area of improving male health using and addressing the antioxidant state of males is a very confusing, but there is one comment that's been used quite a few times, and that was one way to increase the number of good or low damaged sperm is not to abstain from intercourse for too long or ejaculation. Several studies have reported better sperm quality with repeated ejaculations prior to treatment. Less volume, but better quality. And you need to ask your doctor about this. So thank you for your time. I understand IVF is a very personal journey that involves the actions of many people, and it helps to understand all aspects of this journey. I hope this information may be of value. If you do, then tell everyone else about the service and please subscribe. Thank you.